Hey everyone, thank you for joining our webinar. We are sorry there has been some inconsistency with the time. The original agenda was to be initiated at 10:30, but the invitation seems to say 10 o'clock. We apologize for that, but we will of course begin now. My name is Idris, and I will be hosting this webinar. Later on, Pablo will, of course, join on his part, but we will, of course, proceed now. This webinar is in regards to clean rooms, disinfectants, GMP systems, and water systems, of course. I will take you briefly through Eurofins and a small presentation, and we will take it from there, of course. Just some practical information in the beginning. I hope you all can see the panel for the GoToWebinar. In this panel, you will have the option to send us your questions related to the webinar. We will, of course, depending on the quantity of the questions, be able to answer them within this webinar and gather the questions and the replies and send them in the follow-up email after the webinar. All participants will be muted. If you find any technical issues, you could potentially use the option of raising your hand, but since you're muted, it will not give any advantage using that function. By the end of the webinar, every attendee will receive a certificate. Additionally, you will receive a survey, a few questions, and relate to this webinar. We hope, of course, you will take your time to evaluate the webinar. Thank you for that. And lastly, we will, of course, be recording this. The whole webinar will be available at our website. We will, of course, send you the link for that in the follow-up email as well. So, as mentioned, I'm Idris. I will be your host. Currently, I am working as business manager for medical devices in Eurofins, Denmark. Mainly me and our subject matter experts within the field engage with manufacturers of medical device within Denmark and Norway, with the idea of advising and doing the medical device testing in order to get products into the market. I'm a pharmacist of background and been working with Eurofins for 2 years. With me, I have Pablo Moreno, which has a background of chemical engineer and has been working with process control and validation for almost or more than 10 years, I would say. He is our subject matter expert in the field of qualification, validation of processes and utilities such as gas, water, and HVACs. Just a quick overview of the webinar. We will, of course, have a short introduction as mentioned, but Pablo will get into the new GMP-Annex 1, which includes the clean rooms and controlled environments, disinfectants, gas and water systems. We will probably have 20-25 minutes at the end of the webinar for discussions and the questions you have forwarded. And lastly, you will, of course, have the evaluation forwarded as well. Just briefly about Eurofins history, it was initiated in 1987 in a small lab with 4 employees. But today we have more than 800 laboratories with over 48,000 employees around the world in 50 countries. Eurofins operates in 4 different areas, which of course includes pharmaceuticals and medical device, environments, food, and clinical. Within these areas, we have different laboratories. As you can see in this map, we are really good presented in Europe, US, and some part of Asia and Australia. Eurofins Biopharma Product Testing Denmark and some of the other labs is marked as yellow, as you can see in the map. Within Europe, we have different laboratories, some of them which is excellence centers, especially if you see from the Garvin Island, Netherlands, and Munich, which are competence centers for biochemistry, biologics, and bioassay. We have a France lab, which has the capability of having stability storages, raw material testing, and Milan in Italy, which has competencies in extractable and leachable studies. We do have another lab in Netherlands at the left, as you can see, which is a GMP NMR and XDR lab. Lyon, France, which is really special in typing of microbiology. And our, of course, Danish site in Copenhagen, which is competence center for metal testing. In addition to that, we do have several laboratories around the globe, really good presented in Europe for medical device testing. Within the Danish side, we do have, of course, this process control and validation department, which is handled by Pablo Moreno, and we will, of course, dig a bit deeper into all the aspect which includes clean rooms and so on. But the idea of Eurofins is to have one-stop solution and take care of the different aspect of a facility. Besides that, we do have a professional scientific service, which is basically an in-source model for our clients. Interested in that, of course, we will engage in a more detailed discussion. Besides topics as mentioned, we do have microbiology department locally in Glostrup, which can do almost all necessary biological testing, which is time limited. We do, of course, have several other microbiology services which can be provided by other labs around Europe. We do have chemistry lab locally as well, and in different countries around the globe. Primarily for Glostrup, it's the metal testing, and we do have chromatography, spectrophotometry, and we do a lot of method development and validation as well. For the medical device testing, as just mentioned, there is different labs, and we do assist our clients from the early phase of proof of concept until the release of a certain batch. We can assist with functionality testing in the initial phase, mechanically and chemically. We do have, for the design validation, we can assist with functionality and characterization in terms of biocompatibility and chemical characterization. Of course, we do have, for the manufacturing validation, sterilization and packaging validation, and of course, the sterility and lot release at the end. Thank you for listening. I will get the word to Pablo, and he will, of course, have a short introduction on himself, and then we will, of course, proceed with the webinar.
Hi, everyone. Thank you for being in this webinar. Thank you very much, Idris, for your presentation. Very nice. Yeah, difficult topic today, talking about this upcoming webinar, because upcoming requirements of Annex 1, because I think it's a topic that is very, I would say, present in webinars. There are many around, so I would say that we will try to cope with providing key information for you to have practical tips here on how to cope when this upcoming regulation will come into effectiveness. So briefly about myself, I'm business unit manager of the Glostrup site in Denmark, and since 2016 and before, I was in a similar position in Italy. I dedicate most of my time to advise, as Cedric said, so clients to try to provide a one-stop solution to monitoring programs and validation. And of course, we are very focused on testing, so we are experts on that, and this is our area of expertise. I'm subject matter expert for critical utilities, which include also the HVACs and the clean rooms, but also clean rooms themselves, and also to a certain extent, I'm involved in cleaning validations. I'm active member of ISPE, and I think this information is relevant because you will see the imprinting of ISPE on what I will be speaking later. I have to say that special thanks to my colleague Catherine Jepsen, who significantly helped on setting up this presentation and developing the topics we will talk today. Topics to develop, let's say there is indication of time in orange. We will try to keep that times, but are indicative. Big topic today is clean rooms and controlled environments, of course, because it's the scope of the Annex 1. So this is where we allocate more time, and we will be discussing more about it, I'm sure. And there are many updates on that, so we will talk about that. Disinfectant, there is one, I would say, big update, and then all the other topics are more ancillary. So I shorten the time. Then gas systems will be deployed in a way that we are navigating through the requirements. And I think that this is also a big update for the new Annex 1. And then water systems, which I don't think there are many updates. Most of the pharmaceutical clients are already, pharmaceutical and medical device clients are already very aware of the new regulation. As a tip, I would suggest if you can keep open the upcoming Annex 1, that you can, let's say, go to the link that you see on this slide, or alternative, you can Google the words new Annex 1 Eudralex, and then as a first option you will get it. It's because then you can switch, or if you have two monitors, you can be tracking, let's say, the different paragraph through the numbers. So you can use the section numbers on it. So it will be very useful for you to get around where is the information located for future consultation. Some quick forwards on this. We are focusing the EU compliance, and we will be talking about things that may be already known for you because the CGMPs are already addressing them. But that's it. That's how it works with updates of regulations. So they come from the CGMPs. Then some keywords and acronyms, but most of them are known, but the biggest one that you need to keep in mind is the CCS, Contamination Control Strategy. We'll be using that a lot. And the others are very known, Array and CNC, OS, OT, and very common acronyms. Let's start saying that the current Annex 1 is having 16 pages, and the upcoming is having 52. So it is, of course, well understood that there are updates and that there are many new things developed in this document. And it's not an update of the same document. It has been totally, I would say, rewritten. So I think that it has been a fantastic update, really. I really look forward for that to come into operations, because it's really clarifying many aspects of how to handle clean rooms and what is in it. Remember that we will be talking grades A, B, C, D, and C and C, which is a microbial classification, whereas numbers like 4.8, ISO 5, ISO 7, 8, or 9 are, I would say, particle classification based only on particle concentration or not microbial. Let's start then. Let's start, let's say at the first that remember why we are using, it's important to remember why we are using clean rooms, and it's because we want to control microbial and pyrogen contamination. So the focus of this annex is that, let's say. And we need to do that because either we need to control bioburden or we need to do aseptic processing. And if we need to control bioburden, it's because we can do a terminal sterilization, and if for a sterile product, of course, and if it's not possible to do terminal sterilization, we are constrained to use aseptic processing, which is a very invasive way of producing. So the scope of this annex has been, I would say, fully clarified, and I really like this because it is stated that they can, so sterile producers can use as a reference this document, but not only. It might be used for support the manufacture of non-sterile products. It is implicit in the current Annex 1 data already, but here has been further developed, and I really like this approach because many of our customers are saying, why should I comply with Annex 1, which is a document for sterile producers? It is the best reference we can get for control of microbial contamination. There is no doubt on that, and it's a European document that we can really use for our European facilities. So I do believe that this is a very good document for us to refer to, and I do believe that this is an absolute reference that we can take for compliance and state of compliance over the time. Let's take a quick look on what ISO is, let's say, because they are referring each other, the documents ISO. But ISO works to technical committees, and specifically the one for clean rooms is the 209, and they have currently 17 standards. And from those 17, you have the 14604, who has, let's say, there are 14 related to particle classification and methods and how to determine them. And they are extremely focused on the physical qualification of clean rooms because they cover, they cover two types of industries. Not only the pharma and medical device, but also aerospace and micro components. So this is why these documents are really focusing in the particle classification, because they try to be broad enough to cover all that type of industries. And the pharma and medical device industry is the, I would say, regulation, the industry that is regulated by the patient safety. And as a consequence, of course, these technical committees have liaisons with the medical device committees, for instance, with the group of 178, for instance, and so they regulate, let's say, in this way, the interrelationship between committees. But the committee dedicated to the clean rooms is working on standards for many different type of industries. Okay, so that's why I do believe that the reference to the Annex 1 is a very important one whenever we are developing documents that relate to contamination, microbial or pyrogen contamination control. You may say, okay, but what about the 14698, which is an ISO dedicated to microbial biological contamination control, but is so generic that I think that still the reference should be the Annex 1. So actually you can see that they have the three documents you have here states general principles, evaluation and implementation of methods, and evaluation and interpretation of data. But they are not laying down specific requirements like specifications per type of production and that kind of big requirements that we do have in Annex 1. Let's move on and let's start with the first big introduction from the new Annex 1, the upcoming Annex 1, which is the concept of the contamination control strategy, the CCS. The CCS is the, I would say, requirement from this regulation to have everything contained under I will call this like a validation master plan. So a big document that really deals about all the possibilities that we may have to better get state of control in our clean rooms and ancillary services like critical utilities. So this introduction has been, I would say, expanded and improved, I would say. I do believe that this CCS will be, or is already for some companies, deployed in two ways. When you have clean rooms that requires only control of bioburden, so we are talking about the grades C and D or C and C, even the CCS could be a risk assessment of one document, let's say, which may be quite rather simple, let's say. But when we go into aseptic cores and aseptic processing, things will become so complicated because in this CCS, we need to cover a lot of aspects. And so it is expected to have multiple documents, to have a better structure of it. So I do believe that this is a big change, and I do believe that with respect to what we have in Annex 1, which is the request of a formal risk assessment, this is the requirement for today, so this is mandatory for everybody who is, regardless of the class, we need to have a formal risk assessment for our clean rooms to understand where to allocate sampling points, for instance, or stuff like that. But in the upcoming, we have the concept of the CCS, which is, I would say, an expanded version of the risk assessment. That is very expanded, because if we go to the document, throughout the document, it is mentioned 42 times on top of this first introduction in the section 2.3. And on top of that, the list of requirements is quite big. So here at the point 2.5, at the section 2.5, you can see what should be into this document, and it's actually quite comprehensive and should consider all aspects of contamination control and life cycle. This is another very nice and introduction from the upcoming Annex 1, which is the concept of life cycle that well is already widely applied in the CGMPs, but in the current Annex 1 is not that well explained or that well addressed because it's from 2009. So that's maybe. So implication from this contamination control strategy documents is that we need to state on there, like in a validation master plan, which are our approaches for validation of clean room. And this is because it regards many different aspects. So whenever we have clean rooms, we need to match the map of our operation to the approach to qualification. And this is, I would say, extremely powerful for us to identify where the critical areas are and so on, to have a good and useful document that will be updated over time. So that means that the typical at rest and in operation testing based on risks become a little bit more expanded. And actually at the beginning, we can have a sort of ref 0 of this document in which we, let's say, state which is the overall plan towards a new clean room or a refurbished. And then from the information we gather from all the testing at rest in operation, and we design, so to speak, an approach to validation that will reveal that in the best possible way, we create a Rev 1 of the CCS for us to keep valid throughout the life cycle. What I would say is that this is extremely powerful, and there are many companies that they are already using that kind of approaches, and actually it works very nice, and it allows a very good flow of the documentation with very good understanding of the information we gather from our clean rooms. So this is more the movement towards, I would say, instead of doing one-shot testing for clean rooms, we will tend in the future for sure to do more, I would say, extended testing and more robust testing at the very beginning to gather information to feed the CCS to perform, so to speak, the revision 1 from the revision 0. So I'm pretty sure that will be the trend of everyone. So we will be working a lot on that, all of us. So in terms of gradients and premises, there are some updated, very nice actually. I like them a lot, and there are many of them. They try to cope with the new technologies, and there is a lot of focus on airflow pattern visualization, and I do believe that that's a very powerful tool. And this is also called the smoke testing, and is one of the requirements for grade for ISO 5 and 6, if I'm not wrong, from the 14604, from the ISO from 14604. And actually, it is a very good way for us to understand where our, I would say, turbulence or turbulent flows are, so to speak, creating bags of air that are not properly, I would say, exchanged in our clean rooms, and then we might have problems or not having, I would say, the right airborne contamination over there. So, sorry, airborne cleanness over there, not airborne contamination. So I do believe that this is a very nice introduction from the document, and is a very good expansion you get. In the past, it was 1 page only, and now we have 2.5 pages from that in the section 4 mainly. Then let me say that there is a huge amount of information about barrier technology and production technology in the upcoming webinar, in the upcoming Annex 1. And it's really significant, the amount of information you have there. Is mainly to cope with the new technologies, of course, available, but it's not scope of this webinar, so we are not developing any of these topics here today. So if you have questions on that, you can reach me on email or something, but I would say that let's keep the focus on the topics we have to develop today. Let's go to the qualification of a clean room, which is, I think, one of the best updates we got from this document, which is the separation between qualification requirements and monitoring requirements. In the Annex 1, in the current, we have everything mixed up, and I can tell you for sure, from the Dobser, our clients, from a benchmark perspective, all our clients are really, I would say, struggling with that. So yeah, but where shall I apply the same criteria, action limits? So it is really problematic to cope with that, and now has been really nicely clarified. So basically, you can see that these are requirements during qualification, and one of the nicest things we have here is that they put, so the technical committee working in these updates put a lot of focus on alignment to the ISO 14604, because at the beginning, during qualification, our main scope will be to get compliance to the 644, so 14604. And it's a very nice thing that is also claiming that we are not testing for particles bigger than 5 microns, And there are good reasons for that, because the ISO committee thinks that particles are not statistically representative because the limits are too low. If you can recall, the numbers here are very low, around, I don't know, you typically get, if you do a qualification of a new clean room, you typically get 5 or 6 particles, which is not statistically representative. And basically they are saying, well, you can't consider, of course you consider the 0.5 particles because they are statistically representative, but you could move to the next size, which is 1 micron, to get another idea. But what I really like it is the fact that they're keeping the 0.5 and 5, because there is a clear understanding that as many of you may know, that the 0.5 particles are the ones representing the small ones that will never sediment. They will never go down to the surfaces. They will always go through with the turbulence of the air. And basically that's why you have the 0.5. And the 5 is because they go down. It's because we can catch with, passive devices like settle plates, and apologies, and they are very important for us to understand how the air contamination could end up in surfaces. So it is a very good thing that we are keeping them for the high-grade room quality. And you can see also that they put, microbial contamination limits during qualification. We were not having that in the annex. We are not having that in the current Annex 1. They are keeping the same I would say, limits, which is, I do believe that is very good, well represented for the clean rooms grades. So I do believe that this is a very good reference for us. Just for information, in terms of benchmark, which is called the ISO 9 or the CNC areas, which comes after the grade D, is having typically a specification of 300 to 500 CFUs per cubic meter in the air. So it is the next level that is not displayed here. And of course, CNC is something open, and so it's not regulated by neither by the upcoming Annex 1, but I would say that we need to keep focus on that. Okay, so CNC areas can also be very well represented by these qualifications. At rest and in operation is still a requirement. The air velocity, I think that is a very nice paragraph for some of the producers that are struggling with the biosafety lab banks that are working typically at low speed because for biosafety reasons, they typically work at 0.36 meters per second in the unidirectional area. And this is a clarification very nice, which is say that we can, let's say, scientifically justify the CCS this, and without trying to push the limits of the speed, because then we will jeopardize the biological safety. So it's a very nice thing we have this now, because has been, let's say, a reason of many discussions in the past for many of us. What we need to comply in Annex 1 and the regulation of biosafety lab cabinets. The test, I think this is very nice that we have tests now all of them very well described, and they of course are willing to set up, I would say, the test of microbial airborne and microbial surface contamination, which is, let's say, part of any qualification process. And they are referring specifically here to Annex 15. So we should, of course, consider all the CCS approach to the requirements from the Annex 15. So we need to always keep that in mind for us to always get both compliance. Of course, this is implicit but not mentioned in the current Annex 1, but everyone has the expectation of that. So the Annex 15 is the B model of qualification, and we are expected to fulfill that requirements. And here we have very clear expectations towards the compliance to the ISO 1444644, which is exactly the same. This is big news. So the one is now, this table is, let's say, big news because now this is more ISO-like, because the ISO 14644-2 is telling which tests need to be done for continuous compliance of the clean rooms, and this is sometimes called the qualification of the HVAC, because we do the integrity testing, the airflows, and the verification, but also they are here stating if we need to do it for requalification, and they are giving us, which is beautiful, finally a periodicity for this. So we have a specific input for how frequently shall we requalify our clean rooms and according to which testing. So this is alignment to ISO 14644, okay? And of course, they are introducing the micro. Let's move now to monitoring. So in monitoring, there are, as I said before, they are split now. So we have requirements for monitoring, and they address it very nicely, I think, in one simple paragraph there, that we should keep the focus on the reliability of the methods. Why? Well, because actually for microbial monitoring, the technologies may vary a lot. So for particle counting, we use, let's say, light scattering technologies that are very robust and very well determined, that basically they are not impacted by, for example, dryness of the air or other things, or they are known to be, let's say, less impacted for that. But instead, for microbiological methods, we have many things from the production itself that may impact the usefulness of the data we will gather from the monitoring system. And we could be doing a lot of things during the sampling of the monitoring process, and getting false negatives all the time, which is perfect. Okay, get a false negative is said, okay, it gives you the impression of that everything goes very well. But if this is challenged or pointed out by authorities, this will be a huge problem, because we will be obliged to revise data backwards, and that could be a strong headache for everyone. So to challenge the reliability, or to discuss about the reliability of the methods within the CCS, it is a very nice approach to be doing this. And which type of culture media do we use to growth, in case we're using the typical growth medias, which are the pros and cons of not using specifics, and that kind of stuff. I think that need to be addressed in CCS, and I do believe that this is something that is bringing to light a potential problem that we might have. This is not a big one for the typical monitoring of clean rooms, but we will see on the gases that could be big. So we will look into this. The monitoring of procedures. The monitoring procedures should define approach to trending. This is another very interesting and nice thing that has been incorporated, because I do believe that we do, all of us, trends according to the data we have, but it's not defined beforehand. So by defining the approach to training, we will be challenged on which is the best way to gather data. And I do believe that this will I would say if we start all of us doing this in a CCS document, that will, I would say, bring significant improvements to the whole process of assessing the data that we should gather during a qualification process or during a monitoring. So both. So it's a very important, I would say, introduction. This one. Changes in microbial flora, type and numbers, and predominance of a specific organism is part of what we should be taking into consideration, and this is because something that is tied to life cycle, and this is the specific requirements of doing identifications and doing as much as we can, doing a very good investigation of any alert level, any values beyond alert level or action levels. So this is, I would say, mostly implemented for most of our clients and producers. But what I would say here is that it really needs to be incorporated in the contamination control strategy, the CCS. Now we see the limits for non-viable contaminations, meaning particle counting, and now you see the numbers that are limits for the at-rest. And you can see now a limit of 29 for the particles 5 as it was before. So 5.0 micron particles are having a limit for the monitoring part, and this is because we are very interested to know for that particles that may sediment, let's say how many we have. The in-operation, this is an interesting topic here, that the in-operation limits could be settled in order to establish more stringent approaches than this document, based on what type of activities we do in that roles. And here you have the addition of the action limits for viable particles, so for microbial monitoring, and you can see that the values has been maintained with respect to the current Annex 1, and they are, let's say, addressing in the same way contact plates and glove prints. So the fact that this is in tables implies that we should keep good, I would say, monitoring of the surfaces as well. And this, I would say, is all yellow, this paragraph, the 931, because I think it's a clarification, very important, that is part of the CGMPs already, but how we deal with investigations of out of trend OS or OTs is extremely important, because the repetition of the same colonies, despite we're not going beyond values or beyond certain action limits or alert limits, may be an indication of adaptation of organisms. And you might think that this is not common, but I can tell you that is more common than you think. So we have seen them, and we have seen efficacy testing, sorry, for disinfectants failing because of adaptation of organisms. So it's something to keep in mind. There is a big pressure from authorities in this very moment to keep a good eye on this. It is saying that consideration should be also given to the identification of microorganisms detected in grade C and Ds, because there are some studies demonstrating this, that mainly is there where when you have more flora and you have more organisms able to adapt over there. So it's there where it all starts, so to speak. It's where they start to create this thing, and then sooner or later we have the organism within that aseptic core. So I would say that this is a very important thing for us to keep in mind, and we will depict this. I will depict this very well with the efficacy testing of disinfectant that comes through. Qualification of personnel. I think that this has been written. What is typically done already, which is, okay, sufficient and appropriate personnel, suitable, qualified, trained, experienced people should go into our clean rooms, of course. And this is for a good reason. About 95% of the contamination in a qualified clean room comes from people, from movements, from people introducing during dressing, so gownings and gowning qualification is an extremely important activity and is defined in the glossary that the gowning qualification is needed in a periodic basis. So, and you may ask, okay, but is this something required also for the grade C and D? Well, we need to sort of consider that into the contamination control strategy, because of course we will be having gowns that we may not be sterile, and but we need to assess still which is the load of contamination that is existing in that gowns. So by doing gowning qualification at that level, you will understand that, and you can actually identify colonies to better understand how, let's say, this type of organisms that are carrying the gowns can propagate into our clean rooms or controlled environments. And here is a very important, I would say, update that you can see now, the red circle that is saying that unqualified personnel is not allowed to come into the aseptic core, and you can see that regulatory inspectors will not be allowed according to that unless extremely well justified. Let me say that at the same time, the document is saying that in design specifications of our clean room, we should have a very good visualization of the aseptic core. So from now onwards, what an inspector will expect is that you have a very nice window for you to look and stay probably for hours from a non-classified area or a CNC area, looking at how the aseptic production happens, because this is a very frequent requirement from inspectors of aseptic processes. This is not a big one for bioburden control, but is a very big one for aseptic processing. So the glass walls, let's say, will be more and more, I would say, required by inspectors if they are not allowed to go into the clean rooms. That's natural. The concept of disqualification of personnel, this is also very important, and this is because there is very frequently this problem that somebody not passing or having some issues with, I would say, investigations in relation to exceedance of microbial limits. They are still allowed to work, and then well, here this document is clearly stating that we should keep an eye on the disqualification of personnel in case it is not reaching it's not reaching the requirements stated from some of SOPs requiring, let's say, work in aseptic conditions, which is difficult to evaluate, of course, but it can be done through adverse trend or something like that. So repeated exceedances from the same person or stuff like that. Is of course a very difficult topic to address, but I would say that the CCS also should be clearly stating this throughout the qualification of counts. Personal hygiene, cleanness are essential to prevent excessive shedding of, and as a consequence, introduction of microbial contamination. So I think that we need to really keep an eye also on that, and to have engaged personnel working for having always keep that thing in mind. Also the limitation of people that may have other, I would say, may carry, because of the type of work they do, other contamination is also very important and very well addressed in the upcoming times. The inputs on garments has been, I would say, a little, but I would say relevant to be mentioned here. You can go through them from the text you are seeing. And I would say that the fact of broken garments has been broken, or it says, it uses another word, I think it's okay, integrity of the garments is the word that is used. It has been addressed here, and this is of course something to keep into consideration. The fact that all outdoor clothing is not allowed in changing rooms has been before addressed by many risk assessments. So this is something very common. I would say that we should not be that focused on that. There are some little additions to the garments by class the goals per grade of contamination, and there are a lot of focus on the of course something that is already doing, like using of goggles, but the fact that skin shall not be exposed to grades A and B. That's very clear for everyone now, and in grades C and D, what has been stressed is the fact that the shoes should be disinfected or autoclaved, which is also very common to see already. So, and then the gloves this is a nice small paragraph that how the CCS should address the use of gloves, because in some operations in Class D, in Class C, the gloves are typically mandatory in use, but in Class D, there are many classes D or C and Cs without use of gloves, but it's a very important barrier for us to implement if we want to avoid human contamination. Of course, we also need to consider the comfort, and when it's really necessary, but it's a very important point to address. So the CCS should also address that. We're moving now to disinfection. So I would say that disinfection has a very important introduction that we will be addressing in the next slide. And let me first address a typical confusion of terminology, which is the use of let's say, or the mix-up of cleaning validation and disinfection validation and disinfectant validation, and many terms that I use it indistinctly that I think that or interchangeable, but I think that it's very good that we all understand that disinfection only addresses the fact of the attempt of removing, let's say, microbes from a surface, from a given surface that has been already cleaned. So the disinfection and eventual disinfection qualification or validation is not related to the cleanness of the process of cleaning, so to speak. Of course, that will be heavily impacted by it if we have dirtiness, but the aspects of cleanness and cleaning validation are addressed by the Annex 15, and they regard mainly the concept of carryover or MACO and that. So it's not scope of the Annex 1 to define that, but what they try to do in this version in this upcoming Annex 1 is to provide a better definition through the definition of decontamination that you can read here and the definition of disinfection. So how this comes into the picture when we have, let's say, when we have this into the new in the upcoming Annex 1, it comes into the fact that it is required that for disinfection to be effective prior cleaning, disinfection should include a periodic use of sporicidal, and cleaning programs should effectively remove disinfectant residues. And this is a very important thing that was not before, and now we have the requirement of disinfection validation, which is a very big thing, because actually we cannot, the validation represent the spiking of something for us to demonstrate, so that a challenge that for us to demonstrate that from the challenge we can get the scope we want. And basically this cannot be done in a clean room, so we cannot spike a clean room to get what we want. So what is typically used to address this is a three-step approach, which is based in initially demonstrate the fixes have very good and scientifically sound proof that the efficacy of the disinfectants we are using are effective towards the surfaces, all the surfaces that we have in our clean. Secondly, the second step is to qualify the procedure of disinfection, which will not address the cleaning, but indirectly it will. But this is typically done also during clean room qualification by testing at rest. In the testing at rest of surfaces, we are demonstrating if we have an issued procedure for the cleaning, for the cleaning and disinfection, we will be demonstrated through the microbial testing of the surfaces that we can cope with the natural load of the room, and so the procedure, the SOP that we have is effective. And then we have the continuous process verification that will give us, let's say, the validation approach towards that. And how does it work exactly? I will be showing in the next slide. But let me make a clarification here. This is not applicable for the fumigation or vapor disinfection like VHP. Why? Because in VHP, you have the possibility to challenge the process by putting the bioindicators. So basically, you can have a faster validation of it. But in a clean room, if you apply that, you may have, because there are some clean rooms today that have in the whole clean room the VHP, but there has been proven that are challenged for the HEPA filter. So basically, it's something that is not that common. So then the 3 steps mentioned above is the right implementation. Let's take a look at that. So it works in a way that at the beginning we put a focus on disinfectants. At the second step, we put a focus of disinfectant plus SOP, which is the qualification. And as a third step, we keep a focus on how robust the whole process is, the whole process of disinfection is. And then we eventually go to challenge the disinfectant in case we see adaptation of organisms or something like that, which is the life cycle concept. Let's go one by one. Basically, the challenge towards standard organisms is typically done in this phase, in the step 1 of 2, where we are testing what happened with when we spike in coupons of different materials, and then we try to disinfect. We spike organisms, of course, standard organisms, and we try to disinfect them. So this is called the surface challenge, and this is made in a lab. This is not made in the clean room, and from that we get the information if the efficacy of the disinfectant is appropriate or not. That tracks the logarithmic reductions, both for bacteria and for fungi. And something similar can be applied also for the removal, but this is more on the cleaning side, for the removal of pyrogens, which is way more difficult thing that is not addressed here. Yeah, I must say that most of the disinfectant vendors have already some surface testing done on the disinfectant. So they go to the market already with I would say efficacy testing, and they are typically related to the AN 13697, which is the European regulation that allows the producers of the disinfectant to go to the market. But then the US chapter 1072 goes beyond this and try to apply this in GMP concept with the life cycle concept we saw before, in which it requires the efficacy testing towards endogenous organisms to determine if the disinfectant is effective or not. The 13697 is a test against standard organisms known to be resistant to certain chemicals, and so this is why they are standard and reference organisms. The US chapter instead put more the focus on use on endogenous and other aspects as well of the efficacy testing. The disinfection qualification is once I have the efficacy tested, so I'm sure that the disinfectant is effective for all the surfaces I have in my clean room, I can go and, let's say, do the address testing 3 or 4 times, or a representative number for me to be sure that I'm robust enough in my process of disinfecting to say that I can, so to speak, qualify this. In this step, we are not spiking, we are not adding on purpose any type of contamination. What we do is to document that after some consecutive cleaning, I'm getting what I expect, meaning results below other limits or values expected, let's say. And that's stated in protocols, and this is typically done in address qualification. So that will qualify the cleaning plus the disinfection SOP. Please remember, I will emphasize again this because it's very important, that disinfection qualifications are not disinfection validation nor cleaning validations. The cleaning validations is Annex 15, the disinfection validation is after the full process of continuous verification that come now. It means that a qualification, and this is just an example of how a qualification of disinfection could be executed during a qualification of the clean room. So I hope you can follow my pointer here. At the beginning, we do all the testing according to 14644. Then we do a cleaning of the room and we do the classification, which is the particle counts. This is already a PQ for us. And then on the microbial PQ, this is the particle PQ and this is the microbial PQ, we do the test in 3 rounds, and this is just an example, with 3 different disinfectants in case we have a rotation for that. And basically in this way, we are testing, let's say, the surfaces 3 times to demonstrate that I'm robust enough to say that my procedure is robust. And basically, you can also rotate the personnel is doing if your procedures are typically manual. So it's important to address also through the personnel test. All this is very important to be addressed in a proper context, and the context is given by a protocol of qualification. So it is a very important thing to address, not only in CCS at this point, but also in qualification protocols as a scope of the qualification. If we move on to the disinfection validation, that the validation assures that the sterile, aseptic, and non-sterile manufacturing environments are under microbial control. So this is the long-term or continuous process verification that will give us a comprehensive knowledge of how good we are at keeping out from the aseptic core or from the clean and controlled areas the contaminations. And in here, the management of endogenous identifications are key. And this is linked to what I mentioned before, to the fact that we should be, even though we are having contamination in class C and in grade C and D, we should be having a very good, I would say, overview of which type of organisms I'm growing every time. Because if they are all the time the same organisms, we should ask ourselves if we have a sort of adaptation of those organisms. If there is a suspect of adaptation of microorganisms, the initial efficacy of the disinfectant must be challenged. And this is linked to the life cycle approach. So we need to start the full process again, just to understand that we are still in control. And believe me, when we find that organism and we go and do the efficacy testing, we can see many times that we get a confirmation of adaptation of organisms. So it's not a rare thing. It is something that may happen. Of course, that depends a lot on how often the rotation is happening. It depends a lot on many things. I would say that as we are normally operating, that we trigger investigations every time we have OS or OTs. This is a good mapping. That works very fine. But I do believe that we are in a situation in which we should put more concerns towards the whole thing, because the disinfection validation is a long process, and to be good at it requires a lot of knowledge from the process. So remember, these are manual processes. The SOP of cleaning disinfections are typically requiring manual work, how I'm scrubbing the surfaces, and that kind of stuff that are difficult to address. So focus on this is very important. Okay, so we are now in gas systems, and this is part of a brand new section for the Annex 1, because in the past it didn't exist, which is the Section 6 utilities. And basically, the whole section is new. Is introducing the concept of clean utilities, which is used a lot already by many industries, not only by pharma, but pharma medical device. But here again, we have the introduction of the utilities into the CCS. So how, let's say, gases, vacuum systems, water systems are introducing contamination into clean rooms is an extremely important thing to be addressed into the CCS. So we need all to understand this, how it works. So basically, we have here in the paragraph 6.2 that it is considering at high risk this list of things. We have here the water, the gases, and the steam, but we also have all the contact materials and contact surfaces that comes in contact with the product. So we could have a direct impact from the fluid, let me call this the fluid, water or gas, touching or going into the product directly. That is very frequent, I must say, or it goes indirectly through the equipment, through the walls of the equipment, because I'm using a gas to dry a reactor or to dry a surface, and then basically that comes in contact with the product. So I really need to address a strategy in the CCS for that, and it is a good, I would say, improvement also this. The critical quality attributes of high-risk objects should be subject of regular trend and analysis to ensure that the system capability remain appropriate. So what we see here, that is, we apply the same concept of trending on defining how to trend in CCS for the environmental monitoring into the utilities, which is normally done for water, but I don't think it's done for gases. I really do not see that very often. So it is something that need to be addressed in a proper way. Records of utility installation should be maintained throughout the system lifecycle. This is a very important thing for the GMP retention times. Either electronic or paper need to be most likely kept for more than 10 years. Because this is a clear requirement for the FDA, but now it's a clear introduction here, and it's really going beyond the typical 10 years used in pharma market. For medical device, I know that GMP or GLP records need to be kept for, in some cases, up to 30 years for implantables and that kind of stuff. So in terms of data integrity and data retention, I think that medical devices are already beyond the 10 years. So that could not, or maybe not, a big problem for that industry. And this was the only paragraph we were having before on utilities. So on the Annex 1, here's where you can see that this is all brand new, because basically in the past Annex 1 was claiming to have the pipes and ducts out from the clean room, unless justified, of course. But yeah. So From new sections of utilities, we have this paragraph. I think that this is a key paragraph because I do believe that this paragraph 6.18 introduces the concept of the chemical, particular, and microbial quality that we should keep for the gases. And the gases that are in direct contact with the product, or the ones that touch the equipment that will touch the product. Here there is a big, I would say, gap that I see very often, in which, let's say, the gas are tested like technical gases, so without too much concern towards the GMP testing. And I would say that from this document, it's very clear that the GMP testing is required, and is required to be linked to pharmacopoeias and not to the ISO, which is the typical case of compressed air, for instance. Compressed air is very often tied to the ISO regulation in regard to cleanness of the compressed air, actually, but it's all physical testing and content of oil, and that's it. It's all based on that. There is no other concerns towards all the chemical impurities or particulate or microbial. So, sorry, there is a concern for particle, but not microbial, neither other chemicals. Also, there is the next paragraph is stating that we should keep in mind for the gases that are filtered for 0.22 micron filters, we should have at the point of use monitoring programs for the microns to keep, let's say, the point of use under state of control because the requirements of it. So let's try to understand here which are the critical attributes that we are required to fulfill, because they are tied to the pharmacopoeias, as you can see. So we are now going to the pharmacopoeias, and we are looking at which are the typical requirements for which are the mandatory requirements for the nitrogen and the carbon dioxide. And what I did is to put a comparison between EP and USP, because it's very clear for this that it is, I would say, the approaches we have in Europe are way more restrictive than the ones in US from the United States Pharmacopoeia for gases. And so this is for you to clearly understand they are not typically aligned in these two processes. So in terms of purities, we are not here required to look at the purity. So we go to the impurities, and the typical impurities for nitrogen is CO2, so carbon dioxide, monoxide, and oxygen, which matches with all the concerns that producers have, because typically if you are having I don't know, vial inertization or reactor inertization, is because you don't want the oxygen from the air, typically because you have a highly oxidizable product. And so the concern, the limit of 50 ppm need to be addressed and need to be kept under control. It is, of course, that if you are producing the gas on site, you are required to have this control more often. But if you are buying the gas, you can rely on your qualified supplier. You need to qualify it first, but then you can say, okay, but I have a qualification and I have a certificate of analysis from my supplier that I will rely on, but then you need to monitor from time to time. So you can extend the frequency of monitoring for that. But basically, that's the approach. Carbon dioxide is also very seldom produced on site, so this is related to the cylinders of carbon dioxide or big tanks. So what basically relates to the same as I said before. At all of this, you need to add the particle and microbial viable, so non-viable and viable state of control. Which is also required. So you need to have a method that will reveal organisms from your compressed gas, and you need to have a method that will reveal particles without compromising the reliability, as we said before. So the methods need to be so at least verified, I would say. In this case, there is a lot of concerns from authorities because they are always checking that these methods are really qualified or validated, because it is well known that the nitrogen or the carbon dioxide for the content of water they have extremely low. They really challenge the growth promotion of the plates. So it is likely that we will get false negatives from our analysis if we are not really validating the method. So I would say that we really need to keep an eye on having this under control and to be reliable on this method. The other thing we can say here is that there are also new methods. So not everyone is having those methods in place. We do develop this, but still, I think that this is a very important question to ask. Okay, always. Well, here you have the requirements of critical quality attributes for compressed air, and here there is something very important to say, that consider that the air may carry water with it, and is common to see contamination of spores and on this type of currents. So it is something to take into consideration, because air is, first of all, produced on site, and secondly, used widely, very, very widely for pneumatic use that might have contact with equipment or product direct. So I would say that we should keep a good eye on that. The contaminants like, yeah, we have seen sometimes CO2, but not the others. Let's say they are, I would say, rather rare as a contamination. So the other chemical contamination is not I would say a big deal, but depends a lot on which is your source of the compressed air. So where the air is taken, because if taken from the same, I would say, technical room where the air is producer, if in that technical room you have, I don't know, other 10 compressors working, then it could be that you will get a significant amount of monoxide or something like that has been compressed into the current. Yeah, it is something to keep in mind, so to speak. Qualification and testing reference. Here, I'm not going into this document because, well, first, there is no time, but there is also a copyrighted document. So this is a quite recent document, and I do believe that it's a very good one. Has been in part developed by some of our colleagues here in Denmark, and I think that it addresses very well the rationales to determine sampling locations for gases, and how we should address the sampling plan on that. It is on testing is not, I would say, that is having 2 or 3 pages on testing, but I do think that it's an extremely good reference for you to keep in mind. So that's why I pasted here in the slide, so you can navigate this afterwards. Typically, the qualification process for a gas system is that you qualify first the supplier, of course, if it's not self-made, the gas, like nitrogen or CO2. And then you go after the land, let's say, what is delivered at the point of use. Are very simple qualifications, I would say. It should not be that, as long we are using reliable testing. Okay, so remember that depending on the pressure we have on the lines, it could be challenging, so to speak, but it requires case-by-case evaluation. And now moving into the GMP water systems, which is, I would say, here we don't have a lot new except the section 6 that I already mentioned, so we'll go a little faster to get to the final discussion I would like to address with you in regard to rapid microbiological methods and the technologies available. So the sections for water has been, let's say, they are already in use. So of course we use the pharmacopoeias, and in this case the pharmacopoeia for water for injection or purified water are clearly determining, let's say, microbial limits and then the toxin limits and the whole thing. So we get from the pharmacopoeia all what we need. I would say typically the particle contamination is not required unless the water will be used in parenteral solutions, and then you have the parenteral rule of that, but it's a very specific use and is very well known, and it's not addressed here. And here what is addressed as a more emphasized thing is the contamination of biofilms, or with biofilms, let's say. And it's because this is the main concern in every water plant, and this is, I would say, a game changer. If you have, let's say, biofilm problems at your water system, then it's a huge problem, because it most likely will be something that you will be dealing for a long time. And if you have a good design of your water system, probably with ozone in the loop, and so or by night when it's not used, and stuff like that, that really improves the I would say the prevention towards the biofilm forming. I would say that this has been, I would say, improved, but not with things that are commonly used. I just stated here that the critical quality attributes from the pharmacopoeias are giving us allowance for microbial contamination, which is in the case of water, purified water, I think it's 100 per milliliter, 100 CFUs per milliliter, and water for injection is 10 per 100 milliliters. So it is or for 200 milliliters. But yeah, you can check in their own monographies. But basically, here we have all the critical quality attributes very well stated, very well known. I don't know if you knew that the highly purified water monography is not anymore effective. So basically, we have two types of water in the pharmaceutical industry. If we are talking about bulk water, of course, if we go to water in containers, then we need to consider many other aspects, but all those aspects are more related to the possibility of leaching contamination from the bottles, and so that where the water is contained. And because the sterilization process is typically thermal, I would say the sterile water in container is really stressed in many parameters that are related to leaching from plastics or from glass or that kind of stuff. So, but I'm not addressing this. So I think it's a good thing that we can rely on very, I would say, very good technologies today to cope with most of these quality critical attributes. Let me say something that is very important at the beginning of 2019 has been implemented by the new pharmacopoeia, a new requirement towards evaluation of elemental impurities, which is an alignment to ICH Q3. And this has been also implemented for the USP pharmacopoeia, and is not anymore requiring the heavy metal testing. So we don't have anymore the heavy metals, but what we have is the requirement to risk assess which metal shall we go after, and then go specifically after them. And then you can say that linking, because this is inorganic matter that we're talking, the impurities from ICH-Q3, I would say that is a fact that the conductivities, very low conductivities, sometimes allow us to say, okay, I should not have any concern towards elemental impurities unless my product is very sensitive to it. And that could be the end of a risk assessment. But still, it's something that I think is relevant because it's new. It's not addressed in the current Annex 1, but it's addressed through the link through the pharmacopoeia, so it's indirectly addressed. Then there is a couple of paragraphs very related, I would say, to what is the production of cold water for injection, which has been, I would say, many forums and discussions and webinars that we have about that. And it's because Europe was not allowing that before, but now it's allowed. And so they are in this, we are expressing, I would say, how we use better the technology that is available in the market. And nobody has doubted anymore that the water for injection product produced by cold systems are a matter of concerns for the quality attributes. So I think that here has been stated clearly that. So then there is this discussion in regard to the filters, vent filters for the storage tank is also a matter of many discussion. There are many technical papers from ISPE on this, and this is because there have been problems in the past with the degradation of these filters. So has been also addressed that type of concept. Here is the expansion I mentioned before towards the biofilms, in which it is a very clear requirement that we include the approach to, I would say, control, remove, or keep out from the loop the biofilm. The best document to address this is the CCS. So it's the contamination control strategy. And for big loops, that should be a document per se, because there are so many things to be checked and understood for the approach to validation. So I do believe that this is a big, I would say, important introduction here, but still requires very good analysis of the technology I'm using, of course. And then there are more, because in the past, in the current Annex 1, we are not having that. Are stressed more the concepts that alert levels should be documented and investigated. And here we need to really be sharp on a quick identification of organisms, because if we have gram negatives, which are biofilm forming, potential biofilm formings, we really need to trigger food reactions. We see, we have been doing this trend analysis for many companies, and we see the evolution in technologies here. Basically, many, let's say, all plants are having the problem of having from time to time biofilm forming bacteria, and the new ones instead are not having them anymore. So that's especially the ones that has ozone in the loop. So basically, it's a very good, I would say, thing that we can count on it. But still, if we don't have the technology available, we need to deal with it. On this, we can get a lot of help from the PAT, so from the process analytical technologies, in which 2 days are available. There is a slide for this. We'll discuss it. So steam is part of water and is part of all the regulations on this. And well, we should should be obviously properly purified, and typically is produced by how to speak, separated pure steam generators, and the fact that we should clearly define the endotoxin levels on that regards some of the ISPE concerns towards, let's say, the possibility of dragging, let's say, endotoxin from the boiling process. So this should be qualified or validated, but it's a very easy and simple validation that most of the producers of PSCs, pure steam generators, are already addressing in a standard document. So it's something quite covered. A suitable sample schedule should be in place to ensure representative pure steam are obtained for analysis on a regular basis. This is very, I would say, common, and this is already in place for most of the producers. So the reference In these cases, is the same. You can see on the left exactly the same document I mentioned before for the gases, because it's also applicable for the water and steam, and also a document that is a little bit older, because that is the document that states what is represented here in 3 phases: the qualification approach for the water systems, that is the 3 phases approach, in which the phase 1 is the phase to test, so to speak, the automation and all the limits regards to operation of the system and how things are working when they're all put together. And this is not applicable when the system is off-the-shelf system. So for small purification system, this phase could be easily risked out and say, well, I don't need the phase 1. Phase 2 is the one that tests I would say the critical attributes towards the use. So we call this the intensive phase typically, and you mimic the production use, which is typically daily. And for 2 or 4 weeks, you analyze the parameters to show robustness towards across the different quality attributes we have. And then we have the long-term compliance, which is over a year, and this is to determine which is the seasonal variation in the water. And so it's an important thing to keep in mind, but this is an ongoing qualification process. So we reach the point of a quick discussion of what about rapid methods then. Okay, here I think that I do believe that the technologies are already here. So as many other regulations, the upcoming Annex 1 is already introducing the topic, is this paragraph. And it says that, of course, that automated microbial monitoring methods may be adopted after validation has been demonstrated their equivalency or superiority to the established methodology. And here there is a trick, because of course this is something very difficult to reach. So to say that a rapid micro method will be the same as the typical CFU methods, it's a still, I would say. But despite of that, I would say the technologies like the biofluorescence particle detection are very effective for many fluids and available for many types of fluids already, not only air. So we can do real-time detection of viable particles in the air, in the water, and in gases as well. Because with at-site, let's say, sampling methods, we can get that on a pipe, and from that we can get on a machine. And it's because this type of method do not require any preparation of a sample, as long as it's a pure matrix, and basically discriminate between the particles that are passing through a light scattering sensor. Let's say with this, if it's viable or not according to parameters of BFP, biofluorescent particle detection. The methods are already available, as I said, and they are extremely powerful. Try to imagine working in an environment that you know that you have a particle falling down and it's a viable particle. So you stop your production, you wait a little till that particle goes out, and then you continue production. Same could be for water, same could be for gases. And it's extremely powerful. It's so powerful that there are many companies already selling that type of technologies. Many water producers are already including this into their well, I would not say standard, but it is, because it's expensive, but it's as a feature that you can have for your water system production. And it's based on this technology. And I would say it's already here for process control. So for process control, we do not need, I would say, to be extremely picky on the part that is equivalency or superiority to the established methodology, to the CFU. And there is a trick here, of course. The pro is that this is extremely powerful because it enables the process analytical technologies, and so it fosters every decision-making process and really gives you the possibility to be proactive into your plants, let's say water, gas, or clean rooms. And basically, this is because you can react immediately as soon as you can avoid biofilms. You can a lot of advantages. Which is the con? The con of this is the fact that they can actually not, I would say, provide the equivalency to the CFUs. And there are two important things. The technique itself, the biofluorescent particle detection, is having some problem of cross sensitivity, let's call this. So it has been proven that this technology can detect fibers of gums as viable particles. And then, this fluorescence is got to the detector, and so you get a positive. But it's still a particle, and it's still something that we should at least try to react. And so it's something that anyhow provides some information. But the other part is the one that is also very widely discussed around very many forums, is the viable non-culturable organisms. So there is a concept, or there are many organisms that cannot grow in the typical CFU values that this technology is able to detect. So this represents a gap for us. The two things represent a gap for us to keep us in lack of understanding if it's really viable or not. But I would say that we should explore all this implementation, and the implementation is what is tricky, because I don't think anymore that the technology itself is tricky. The technology is being consolidated in this very moment. So there are many producers already. There are, I would say, many companies that claim for robustness of the machines they have. But the problem at implementation is that we should, I would say, try to be, I would say, more open-minded towards what is really our objective in regard to process control. So basically, we can imagine many different situations, but the example I provided before, in which the person is, let's say, staying away from the production while we have the detection and then comes back. And if this is stressing too much the process, then you cut the technology out and you don't use it. But actually, it is a very powerful gain that we will get for process control. We really hope that this will be back to, I would say, to review from authorities, in which they are promoting one technology over the other, or we really would like to see, let's say, position papers in which you say, okay, I am a big pharmaceutical producer, my technology is that one, and from that one I will determine sterility or not of my clean rooms based on this method. And the comparison may be, I would say, monitored forever. So we should still keep an eye on the traditional methods while we are testing with the automated and rapid systems. But still, I think that this is a big gain for every one of us. So this final discussion is, probably you will have a lot of questions about this, but this is emerging technologies, and believe me, there are many forums. I can provide some more information about those forums, but you can actually see that this is a very hot topics all over. So let me go to the take-home messages. So I'm wrapping up here the things, and I use these symbols to say what is good or what is not that good, I would say. And so the fact is that the CCS requirement It's not only a risk assessment might represent a problem for some of us, because in the past, from the Annex 1, we were only required, from the current Annex 1, we are only required to have a document, which is a risk assessment on how I use or sampling locations or something. But now we will be required to include everything, to include the fluids, to include approach to validation, to include. So I do believe that this is, nevertheless, is a very nice upgrade that will create a gap. Okay. Then what I do think that is an improvement to all of us, without constituting a gap, is the separation of clean room limits for qualification and monitoring, because this is clarification, and this is, despite most of us are already doing what is required here, I do believe that it's better to have it in a document very clearly stated, so we all do the same and we are not inventing approaches every time. So I think this is also very good. Here, requirement for disinfection validation may introduce gaps, that's for sure. So I can tell you that this will be a challenge for the ones that are working in control of bioburden, because the ones working in aseptic production, no doubt that they will have good understanding of how the disinfectants are working throughout the life cycle of the clean room, because they need to put a lot of focus, because one growth takes out from specifications, whatever. But in clean rooms where the tolerances are higher, I would say it's a little bit more difficult to determine, so how to go to identifications and so go to possible adaptations of organisms. So here, what we need to do is to really try to address this in the CCS, and by addressing the, I would say, identification policy, or an out of tolerance policy, or an out of alert limits policy, to get this in the best, to address this in the best possible way. And then I would say that the new section of utilities also expands and improves the requirement, but also add gaps. And these gaps, I see clearly the gap for gases and monitoring of gases, which is something that I've seen very little so far. And I would suggest to keep an eye for the future of this. Water system requirements are already implemented by most of the pharma and medical device, reference to ISPEC guidelines. This is already in place. I don't think that will constitute a gap, but it's nicely explained and discussed in the document as well. And then for gas systems, as I said before, this represents a big gap, as not implemented yet for most of our what we see in pharma and medical device companies. This is all there is from my side. So I will appreciate if you can just start for the questions and answers. And thank you very much for listening, and then for being with us with all this attention.
Thank you, Pablo, for a very good presentation. We will go ahead with some of the questions you have asked, and we have received several really good questions, but I think we will go for some of the most relevant ones, and once we cannot do in a timely manner, we will of course forward that questions and the answers in the follow-up email. Okay, one of the questions is, are all these new requirements applicable for compliance for medical device production facilities?
Yeah, well, an easy answer to that is yes, because as I said before, the Annex 1 is a requirement. It's a very good and comprehensive document to take into account whenever you are trying to achieve microbial and pyrogen control. So I would say yes, despite you're not having aseptic production, so you have your terminal sterilization, all the parameters for you to do a proper state of control are there. So why not use it?
Perfect. We have a question related to clean room. How do you assess holding during clean room qualification?
Okay, that's a very good question. I think it's well, this is related to the separation I mentioned before between validation of the cleanness, like I would say cleaning validation ruled by the Annex 15, and the disinfection validation. So I do believe that the only way to address holding validation is to challenge the holding. So to say, okay, I need over weekend, but if I have no personnel going in and I have my my clean room validated to low particle emissions from the filters, and they are all, I would say, under control and demonstrated through the qualification. If I'm not having lowering down the speed of the air in the clean rooms and keeping the positive pressure over a weekend when people is not going in, the holding is not a matter of concern. The holding could be a matter of concern if there is a product exposure over long periods or something, but yeah, that should be specifically addressed in a challenge.
Perfect. So more generally, we have been asked if the presentation will be sent and forwarded. As mentioned earlier, we are going to share this recorded webinar in the follow-up email. You will receive a link with the access to our general website, and then you will be able to see the presentation. Again, with these questions, these will be available as well with answers. Another question related to clean room is how often should you revalidate a clean room?
Okay, yeah, this is stated in one of the slides. I cannot recall which part of this, but it's clearly stated now in the upcoming document. It was not stated before that the expectation is to have every year the requalification of grades C and D, and every 6 months for A and B. But also you have a specific requirement test by test, so you have a very good indication now of which are the tests you need to do at every requalification, which is very nice also.
Okay. Related to disinfectant, we're going to take some questions in different sections. Related to disinfectant, do I need to test all my disinfectants? Do I need to rotate the disinfectant for class C and D?
Yeah, okay, that's a very good question. It's a matter of concerns of many that shall I rotate or not? Well, in class D, I would say, and on CNCs, the rotation of disinfectant is not an expectation, and it's not ruled in there, so we will keep like that. So there is no gap on that. In class C, I do believe that this should be matter of the contamination control strategy should be addressed there, because you have many different types of use of the class C. So it could be a productive class C, or it could be a corridor. So if you have only a class C and a corridor and you don't have nothing else, well, I would say you can have let's say avoid the rotation of disinfectants, but still you need to keep an eye on the growth. And if you're having only one, then the immediate implementation is to go for aspergillus.
Perfect. We're going to stick with the disinfectants. Another question, dear speaker, concerning disinfectants. How many batches should be used in the efficacy study? It is a huge work and often difficult to get 3 different batches at one time. Will one batch be enough? or should I use the continuous validation?
First, let me reply to, shall I use the continuous validation? Yes, of course, because we need to keep an eye on everything, but that will be done naturally. The 3 batches approach of disinfectants is not typically used because the AN regulation that I mentioned before, the 13, well, I cannot recall the number, but then we can check on the slides, that enables a vendor of disinfectant to put it in the market already has some requirements towards the products, the variability towards the batch to batch or that kind of stuff. So basically, by testing the typology of disinfectant, the specific product, we are, I would say, safe enough to say that we have efficacy tested. Okay, so all the tests on the formulation so comes before, and those are made by the vendor.
Another question related to disinfection is, are there different qualifications on disinfectant, such as disinfectant qualification, disinfection qualification, and disinfection validation, mandatory for all room classes or only aseptic?
I would say it's mandatory to show that we are in a state of control. So in whatever room class we are. So of course, we are talking about class C and class D, by burden control, I mean. And I would say that it is expected to see that we have evidence of, for class C, that we have evidence of effectiveness of the disinfectants. And the validation will be throughout the fact that I'm not having many, I would say, out of strengths or OS or something, which means to me, okay, the microbial level is under control, so I should not be concerned towards my disinfectant. So I would say the short answer is yes, but the long answer is that need to be stated clearly, risked out in the CCS document or in some documents, clearly stated why we are not going for the full testing.
Thank you, Pablo. Related to gases, when do I need to test gas on the GMP?
Yeah, this is the introduction I mentioned in the utilities part, which says that if the gas is in direct contact with the product, pharmaceutical or medical device, and or is in contact with equipment that is contact with product, then I would say the gas is a GMP gas. It's not a technical gas, I would say. And so we need to fulfill the requirements of chemical impurities control, microbial impurities control, and particle. For chemical impurities will refer to the pharmacopoeia. For the other two, will refer to the class where the gas is. So if I'm having the gas in class C, I will have specification, at least as a minimum specification of that class C.
Thank you, Pablo. We will take a question related to water systems. And we have already a qualified water system, but where is the gap in order to be compliant to this new Annex 1? I think you went a bit around that, but perhaps collaborate.
Can you repeat? So basically is...
We have already a qualified water system.
Okay. The gap? If you need to read it, well, for the water, I would not expect that there will be created many gaps on that. So I would say that should not be triggering new qualifications of data. The thing I will put a lot of attention is on expanding the retention of all the data gathered, because it could be more than 10 years that I will need to retain it to keep the data for me to be GMP and that. So that could be the gap. Yeah.
Okay. Well, let's take a few more questions. There's one: do we have to continue to recalculate CFU per 4 hour for subtle plates?
A good question. Well, actually, in aqueous testing, the 4 hours should be taken because it's the specification. So we are taking. The other hours is, let's say, related to the following of the production. Okay, so if I work in less than 4 hours, I should use less time, and that should not impact or calculate, in theory, a new specification. But if we have a production that will go beyond the 4 hours, I would suggest that we check which is the validation of a method for collection of settle plates, because typically more than 4 hours the plates become dry and say are losing growth motion. That's it.
Thank you, Pablo. Another question is it necessary to validate BFP?
BFP, okay, yeah. Yeah, it is. I would say that this is the rapid micro methods, and there is a clear expectation. But this is if you are going to use the rapid micro method for quality control scope, for process control, for you to make decisions on process control, we need to refer to just robust technology. Okay, so that's why most of the water producers are installing already that as a feature of our water plants, because it's not in the loop. That's not the scope of that is as soon as it is detected that a live form of organism or particle goes into the loop or may go into the tank, it diverts the water to another place, and then it starts working again, let's say, when that disappears. So it is a process control scope. It's not in that cases, you can use without validation, but we need to risk assess, of course. For those that are not familiar with BFP, the BFP is the biofluorescence particle detection systems. So, and they are required to be validated if we use for quality control scope. If we want to replace, so to put to 0 or to put to a very minimum number of the CFOs, measure it in quality control.
Thank you, Pablo. I think we will take around 2 questions more. There is one rather long question or comment. I will just read it out and then see if you can try to sum it up. How should we handle objectionable organisms? Should we investigate all isolates found on agar plates, grade C and D? What should the action be if you find an objectionable organism in an air sample and nothing in the product? How to define objectionable organisms for air for all grades A, B, and C, D. So that's the second or third question, or should it be the same as in product?
Yeah, I think that part clarifies a lot what objectionable organism is as a third thing. So something that should not be in the product because you have a specification for that is an objectionable organism. So the answer is yes, they should be investigated to the full extent, the presence of that organism on all, let's say, the type on all lines. I would say air, fluids, or air and gases or water. What I would also suggest, because this is in the context of investigation, so it needs a contextualization, otherwise it's difficult to talk here. But objectionable organisms are very well defined and very well studied, and then like Burkholderia cepacia is one of them for water, and then let's say they are known to be, there is a manual of bad bugs, it's called for FDA, and so we can get a lot of reference from, I would say, scientific bibliography, in which we understand why it's objectionable or not, and then we need to relate or link that to our product, because of course depends on the product we are producing and the specifications we have for it. So I do believe that objectionable organisms is a big topic, and it should be addressed most likely in the CCS. Yeah, but I would say whatever detection of or identification of objectionable organisms will trigger a very big investigation. Please remember that, I would say, the typical organisms you find out in water systems, gas systems, and environmental monitoring, they are not objectionable for most of the cases. So it is typically the investigation is triggered for the scope of process control, but not for the scope of full investigation into quality control. But try to imagine a product that has, I don't know, an antimicrobial, should have an antimicrobial effect, and then you get objectionable because it's resistant, and then you need to investigate all your products. Yeah, you cannot even imagine. That could be a very big thing. But yeah, objectionable products are actually a very big topic to address.
Thank you, Pablo. I think we will take one last question, but before we do that, I just remind you guys or ask you to please do our survey and the questions we will send you after this webinar. We will appreciate it for the time you're going to use on that, but it will be really productive to have that feedback in order to optimize future webinars or seminars. Last question, I think we're going to take two because it's pretty much close to each other. But the question is, we operate in CNC open area, but we consider Class D requirements. What is relevant for our room cleaning qualification? What should be our CQA? And in regard to that, any recommendations for limits for CNC rooms?
Yeah, well, as you said, you are applying limits of Class D, so we'll take the limits of Class D. And for CNC, I mentioned that typically for particle in CNC is taking the ISO 9 for particle for particles is ISO 9, and for air contamination, so CFU per cubic meter, should be something around 300, from 300, which is a conservative number, to 500, which is, I would say, a more loose number. But I've seen many of those numbers around, and you can do 500 with another limit at 300. That's for sure for CNC. For the topic you said in regard to cleaning or disinfection, I guess that you are trying to address that. What I would say is that D clean rooms or CNC rooms, you address the good, let's say how good your cleaning is through the qualification process. So if at rest testing you are getting numbers that are, let's say, way below specification limits, you can really trust that your cleaning is enough to say, okay, I'm good for it. So it's not a complex process, that, especially if you are in CNC and you're asking for this, so you are already overdoing that. So I do believe that with that you should be okay. I hope that answers the question, because probably I didn't get that wrong. But you can write to me, guys, and just give it.
Yeah, perfect. But when we will send out these questions with answers as well, and of course, contact information on you is available in the presentation as well. So all attendees are of course welcome to contact Pablo directly, and we will, and Pablo will of course be available for any sort of a dialogue related to your specific needs, for sure. I think that's it. Good questions. I think if you have any questions, please send them in because we will have them all together. And yeah, thank you for joining and listening. And we of course do apologize for the inconvenience initially with the time, but we hope that you had a good time with us and this was really useful for you. We know that is a topic that is really relevant in all facilities. Yeah, please let us know if we can assist you further in this subject. Thank you all. Have a nice day and take care.
Thank you everyone. Bye bye.